it's always really helpful to, do your exploration and learn through side projects. And I think research is a very involved process, and it has, a variety of ups and downs. It can be very stressful at times. And so I think the best litmus test to know if this is if you want, how you want to approach biology, whether it's through, supporting startups in the future, through the business side or, funding side, or if it's through traditional sciences path. I feel like the only way to really know is to really, get yourself out there and, get involved with different experiences, both in and outside of the lab.
The Perspective Podcast is a place where I share my conversations with experts from various fields that have incredibly unique perspectives and experiences. We talk about everything from their work, their lifestyle, the unique perspectives they might have, and advice for teenagers like us. What's up, guys? Today's podcast was incredible. I learned a lot myself from Michael, and I hope you will too. We ran out of time in our conversation, and I had about a dozen more questions to ask. So we're going to record a part 2 of this call, and I'm really excited, and I hope you are too. Got any questions for Michael? Just slip that into the comments, and I'll make sure to ask. Have fun. Welcome to the Perspective Podcast, and today I'm very excited to be welcoming a guide, a scientist, a bioengineer, a genius, an incredible person, Michael Trinh. His vision is to apply the principles of synthetic biology to design the next generation of immunology. He's currently an undergraduate research fellow at the Brain Repair Lab at the University of Toronto. He was the co-founder of Nucleate Dojo, a community and training ground for the next generation of undergraduate life science innovators. Michael was also involved in exciting research in the Fanzor family of proteins, which are a part of a new gene editing technology that has big implications on our lives.
Hey, Ditya, thank you for having me on. It's an incredible intro. Thank you. Definitely do not deserve that. I definitely do not deserve that level of praise, but I sincerely appreciate it. It's great to be on, and things are busy, but going well. How about you? How are things going on your end?
Same, it's going awesome. You're somebody I really admire, so thank you so much for taking some of your time to be here.
It means a lot. Thank you. Thank you for having me. I mean, it's always amazing having these kind of conversations. So I'm really excited.
Today, I wanted to talk about your research in Fanzor proteins. But before we get into that, what are you currently researching that you're most excited about?
Yeah. So Fanzors was one of the projects that I was working. It was one major project I was working on over the last year with the Abagut lab. Which is like a molecular engineering lab over at MIT. And one of the other projects I was working on is this RNA sensing platform called radars. Like radar, like the actual device, radars. And radars is basically it's basically a RNA sensor. So like a messenger RNA sensor, something that binds to messenger RNA. It's a messenger RNA sensor combined with a full frame messenger RNA. And so basically what this means is that it's a messenger RNA that will only translate the protein encoded if the messenger RNA sensor is bound to something. So like if you have a marker for a certain cell type, like marker X, if marker X is in the cell, then translate protein Y, attached to the sensor for marker X. And so it's like an if true statement for biology, if we're thinking along the terms of like Boolean logic. It's an if true statement for cell biology. And so this platform was something that the Abagut lab helped pioneer, and I'm currently taking that project that I was on back last year, and now that I'm back at University of Toronto, I'm actually applying that project for use cases in different glia cells in the brain. So we're trying to study, different cells in the brain besides neurons, and exploring their role in neurodegenerative disease, and how we could possibly exploit different mechanisms that these cells use to actually, help treat neurodegenerative disease and reverse their contributions to these terrible neuropathies.
That's actually really cool. The radar system that you talked about really connected to me because it's basically an if-then statement. So hypothetically, you could be able to solve things like diabetes, where you can see what the insulin level is outside of the cell, and based on that, you can produce more insulin. So that was a really cool idea.
Awesome. Yeah. Yeah,
What do you
it's a
think?
crazy platform.
Mhm. Yeah. What do you think is the future of synthetic biology? And also, why is Fanzor a technology that can be a game changer in the synthetic biology space?
Yeah, so two huge questions there. So let's
Yeah.
break it down, right? So
All right.
when we look at the field of synthetic biology, there are so many different applications of the of the technology that we build. Because synthetic biology, at the end of the day, is rearranging working with natural parts and making new parts that nature hasn't created to come up with new applications and use cases that nature isn't doing, or maybe in a different way than nature does right now. So
Mhm.
taking what's out there and making new stuff to create new applications. It's like it's basically the possibilities are limitless when it comes to what you can bioengineer. Um, so when we talk about progress in synthetic biology, you have to look across so many different fronts. Bacterial synthetic biology, it seems like every new week there seems to be, we're putting more and more complicated
Mhm.
things into bacteria and building more complicated DNA constructs in bacteria.
Mhm.
For those in the audience, there's these things called like bacterial artificial chromosomes or BACs. And these BACs, these like enormous like constructs of DNA that house new genes. It's kind of a way for us to test what's possible in terms of like reprogramming a bacteria to do all kinds of new things. So we're stuffing more and more complex DNA constructs and circuits and designs into bacteria every day. So bacterial synthetic biology is going, is a super exciting field.
Mhm.
Um, and then you get into the world of like mammalian synthetic biology, looking at new things like cell therapies, gene therapies, all kinds of really awesome applications in like, medicine. So mammalian synthetic biology is like a breeding ground for like cutting edge therapeutics.
Mhm.
And then you have the genetic engineering field, right? And then, you have technologies like CRISPR prime, base editing, now Fanzors, right?
Mhm.
And so I think protein engineering. So across the field, to give you a broad answer, it's just like an explosion of like
Mhm.
innovation and new papers coming out every week. And so there's an article that's really fun that I recommend people read, especially like students, by Alexei Guzzi. Alexei Guzzi is this he's like a thinker slash like huge mover in the field of both
Mhm.
bio and machine learning and AI. He started this fellowship called New Science, which is like funding grad students. And before he funded New Science, he had a series of blog posts on like how funding in academia works. And
Mhm.
in these articles, he starts off by kind of setting the perspective of how fast biology moves and how fast bio moves. And
Mhm.
I highly encourage people to read it, check out his blog, Alexei Guzzi. Guzey is his last name,
Okay.
is how you spell it. And yeah, it's an incredible read, and it's a credible testament to how fast things move in pretty
Mhm.
much so many different fronts, except funding structures, which is what he was attacking in that article, like how we
Mhm.
get money in academia. But yeah, so future synthetic biology, like booming overall. No matter what field you're looking at, everything from protein engineering to bacteria, to bacterial circuit engineering to cell therapies.
Mhm.
Fanzors, how Fanzors fits into it is it's basically like a new set of tools that are a huge arsenal of like genetic engineering, like proteins that we can use for genetic engineering. So
Mhm.
Fanzors is it's a broad categorization of like basically two related families, Fanzor 1 and Fanzor 2.
Mhm.
And these are basically proteins that are like found in the genomes of both eukaryotes and like the ancient viruses that presumably were infecting eukaryotes.
Mhm.
So these viruses basically, they had Fanzor proteins, and these Fanzor proteins, at least what we think they do, is that they were helpers of the virus to infect eukaryotic genomes. We can get into
Mhm.
that later, but these Fanzor proteins basically help the viruses get into the nucleus of the eukaryotic cell and actually integrate their genomes into the host DNA. And
Mhm.
so, if anyone in the audience is familiar with CRISPR, CRISPR is a bacterial immune system. So it kind of does the opposite. It allows bacteria to like fend off viruses by like identifying viral genes and deleting them and like destroying them.
Mhm.
Fanzer is like almost like an anti of that. It helps viruses infect
Right.
cells, but instead of bacteria, we're looking at eukaryotic cells.
Mhm.
But their function, when it comes to like genetic engineering, we're talking about like gene editing and genetic engineering use cases. Their function is very similar to CRISPR, especially CRISPR-Cas9, in the sense that we are guiding a nucleus, something that can cut DNA using an RNA. So you can specify a cut site in the genome using a guide RNA, and as long as it meets some basic parameters, you can basically guide the Fanzor protein to that site, and it'll cut the DNA. And so that's the 101 of Fanzors. So Fanzor 1, Fanzor 2, they're both very similar families.
Right.
But scars of a virus past basically in our genomes that we can leverage now for doing genetic engineering, as we've demonstrated.
That's super cool. And we'll probably talk about how Fanzor actually works in a therapeutic setting. But for those who are listening who don't have a good grasp of gene editing technologies, the original CRISPR-Cas system was discovered in bacteria. It basically works by destroying invading viral DNA as an immune mechanism. An RNA-guided Cas protein basically makes a double-stranded break in DNA, destroying it. And in therapeutics, a Cas9 protein is induced into a cell. It makes a cut in DNA, and using intracellular repair mechanisms, it makes an edit. So, Michael, what is the difference between CRISPR, which a lot of us are familiar with, and Fanzor in terms of like experiments?
So when you're characterizing a new nuclease, like a new protein that can cut DNA, there's a lot of overlap, I'll say, with how you can go about finding the basics of like how this thing works as a genetic engineering tool. So a lot of the questions that we asked early on are very reminiscent of like something that some of the early scientists working on CRISPR would have asked. So like if you're thinking about this protein that can be guided to somewhere in the genome and then cut it, you have to answer a few questions. Number one, what's guiding it, right? CRISPR kind of gave us a model to understand that these nucleases are guided by RNA. So you have a guide RNA that kind of shuffles this protein, like this nuclease RNA complex, to a certain site of the genome, which you can then cut, like a search function.
Yeah.
And so the question then becomes, what does that guide RNA look like? It's going to be a bit different from CRISPR-Cas9 because this is not CRISPR. It's a different family of proteins. So what does that CRISPR-like RNA look like, or the Fanzor RNA? What does the Fanzor RNA look like? How is it structured? Where in the genome is it encoded? And so, finding basically trying to find what that guide RNA looks like, that Fanzor RNA, it involves like a lot of like genome searching and trying to find like a locus that houses something that you can imagine would be a guide RNA. And then it comes to a process of like, synthesizing your own, building your own, and seeing if that actually works. Does it actually like, complex? Does it form a complex with the Fanzor protein? If it's a Fanzor RNA, it should be able to bind to the Fanzor protein. So there's that question. What does the RNA look like? Another question, which is like, what is the recognition sequence? Sometimes when you're looking at whenever you're looking at like a genome engineering problem, like a CRISPR, for example, whenever you're looking for a site to target with your guide RNA, you have to hit somewhere that has what we call a PAM sequence, or a protospacer adjacent motif. It's basically like a nucleotide signature that is needed for the Cas9 protein to recognize where in the genome it's attacking. It's like a little flag. And we presumed that Fanzor's had something similar, which it does. So instead of a PAM, we called it a target adjacent motif, or a TAM. And so that's another question you have to answer. What is the TAM sequence? In CRISPR-Cas9, it's NGG, N being any nucleotide, and then guanine. That's one protein. Fanzor's is a whole family, so the TAM sequence depends on what specific Fanzor protein you're looking at. But it generally, it generally has to have that. It needs to have that signature for it to be recognized. So that's another question. So there's a bunch of different questions you can ask, right? And then there's a whole like engineering question, like what kind of DNA editing efficiency can we get if we throw this into a human cell? What percentage, if we look at all the DNA that we threw this thing into, what percentage of that DNA is actually being edited? what are the off-target effects like. So it's very, very similar, basically, is the main point to that early CRISPR-Cas9 research and the early, nucleus profiling research. It's a pretty awesome process, a pretty cool process. Very, very it can get really grueling for sure, trying to answer all these questions. But I mean, ultimately, answering these questions allowed us to kind of like, set the groundwork for profiling these proteins. It was pretty awesome.
I'm taking notes as we speak because it's actually so cool. So adding on to what you were just saying, so recently when I was reading the news, I read that Fanzor is considered a game changer because it can edit eukaryotic cells, right? Like human cells that have nucleuses. So looking at that angle, why would Fanzor be better than CRISPR, specifically in eukaryotic cells?
So I'll preface this by saying right now, Fanzor isn't better because it's so new. But you're right. The promise is that, right now when we use CRISPR, we're using a bacterial, we're using like a bacterial or, protein at the end of the day to edit human DNA is like the end goal, right? But imagine if we found Fanzor is an emerging family of research, and it's an emerging family of proteins that we're just starting to understand. And it seems like these proteins are found, decently high up in the tree of life. I mean, another paper on Fanzor demonstrated this thing working in clams. They just like bought clams and like isolated the Fanzor proteins, and they got it to work, do genetic engineering with it. So the question then becomes, can we find a version of Fanzors in like some sort of animal, maybe, as high as the tree of life as like, like primates, right? Could you find a Fanzor that high in the tree of life that works, or get it to work? And if you can, then you have a genetic engineering tool that comes from an animal or a species that's like way closer to us in terms of like relative, like, relatedness in the tree of life than like a bacteria. And that has a bunch of implications for things like, potentially editing efficiency, immunogenicity, if you throw it into a cell, if God forbid you can find a human Fanzor, if you throw that into a cell, for example, there's implications there for like probably the immunogenicity. Something coming from bacteria is our bodies are very good at picking up different signals from bacteria, and so that does become a question you could potentially ask, like, can we reduce any immune risk with that? But in a more practical note, because CRISPR-Cas9 is still an amazing genetic engineering tool, and it doesn't kill us right now when we throw it in human cells per se. But a more practical question one can ask is like, going back to that whole NGG question, or NGG thing I mentioned, like the PAM and CRISPR-Cas9.
Yeah.
If your target doesn't have a PAM sequence, if the place you want to edit doesn't have a suitable PAM sequence, you can use other tools to get that same job done, right? You can use a Fanzor protein that doesn't need an NGG. Maybe its TAM sequence is kind of different. You can target different parts of the genome as well, which is another like just promise in general of having more genetic engineering tools. There's more options that you can use. You're not pigeonholed with like one toolbox or one tool in the toolbox. So overall, having a eukaryotic sourced genetic engineering tool can be very game changing if we learn how to answer some of these questions, and as we optimize the technology more as well. And overall, it gives us more optionality in terms of where in the DNA code we can edit, and do some pretty awesome things with.
And the toolbox you were talking about is, I guess, it's continuously growing every week with new papers and things like that.
Sure, seems like it, yeah.
Yeah. So in your paper about Fanzors, there is discussion about the phylogeny of Fanzor. So, and you also kind of talked about this before. So could you tell us about how researchers found out that Fanzors actually have a CRISPR-like capability?
Yeah. So the early efforts to mine the genomes of like basically the tree of life for like these proteins came about by looking at the domains that actually CRISPR-Cas9 uses to cut DNA. So there was a lot of talk about like, basically predictions on what Fanzors would be comprised of, like what kind of like protein parts or domains these Fanzor family proteins would have. And so our team was looking at one of the domains that Cas9 uses to cut DNA. So for just as a quick refresher, CRISPR-Cas9 makes a double-stranded break at the DNA cut site that it's guided to using two protein domains. Those protein domains being HNH and RUV-C. And so those are the two domains that allow the Cas9 to cleave both strands of DNA. You got two domains to do it. Now, our search was basically taking that RUV-C domain and basically looking for RUV-C like domains in the tree of life, specifically in eukaryotes. Because the RUV-C domain has a clear job, which is cutting DNA. And so something with the RUV-C domain should be able to process and, assumedly process and cleave DNA. And so that was kind of our big searching spot, looking for proteins with this RUV-C like domain. And inevitably what we found was two big clusters of genomes that had this RUV-C like domain. One being in like a bunch of different eukaryote species, things from like different like amoeba to like different clams and stuff like that. And then the other cluster was that in these different viruses. And so these two clusters basically went on to make up the two categorizations that we came up in the paper, Fanzor 1 and Fanzor 2. With the Fanzor 1 proteins, with the Fanzor 1 proteins being those found in the those found in the eukaryotes, like like the different like both like single cellular organisms and also like the things like the clams. And the Fanzor 2s were the ones found in the actual like viruses, specifically viruses with double-stranded DNA genomes.
So that would include things like not retroviruses, like HIV wouldn't be included in that. But I was thinking back to my grade 11 biology class and CRISPR, and thinking back to that. So bacteria was always a precursor to eukaryotic cells, correct? So I'm thinking that maybe the genes that are related to the RUV-C domain maybe even was conserved and is related to Fanzors. Would that be a good assumption?
The story seems to be a bit different when it comes to Fanzors. So an interesting feature of Fanzors is something called the nuclear localization signal, or the NLS. We found this on a bunch of different Fanzors, and the fact that we found this in the viruses we found this in the viral ones is very interesting because so CRISPR, like as we know it in bacteria, is a defense system. It protects bacteria against like invaders. But looking at Fanzors, it really seems like Fanzors was like almost like a part of the Trojan horse to get viral genomes into eukaryotes. So in a way, it's an attack system. It's a tool that viruses use to attack eukaryotes. And the presence of Fanzors in the eukaryotic genomes, at least what we think, is like a scar of like this war between eukaryotes and our viruses. And the Fanzors being one of those tools of war that the viruses use to get in. And so, it's possible that it's very possible that these viruses obtain these nucleases from, I don't know, either other viruses or bacteria. Somehow these eukaryotic viruses obtained, the nuclease, the Fanzor nuclease, and they evolved it. They co-opted it basically to attack human cells, or to attack eukaryotic cells, which are the human cells, eukaryotic cells. So it's a very interesting story, and there's basically the homologue. There's a bacterial homologue of the Fanzor proteins, which is TNPB. TNPB is a protein that helps basically transposons integrate. And so, the fact that these two are related families, and they both help with like DNA integration. It's possible that TNPBs are like TNPBs basically help the bacterial viruses attack. So, attack bacteria. So it's a bit of a different story from the CRISPR side of things, for sure. Which also makes this even more interesting, right? Like it's like it's a sign of the arms race between viruses and their hosts.
In your paper, you talk about the system with Fanzor as Hermes, which is basically horizontally transferred eukaryotic RNA-guided mobile element systems. So how exactly does Hermes work? So yeah, basically step by step, how would a Fanzor protein go to a target DNA and make a cut?
So if you're using it from a genetic engineering perspective, it's going to be very similar to a CRISPR system in a way where you would basically have like a either a DNA construct where the Fanzor RNA is encoded with the protein, or you have what we call a RNP, a ribonucleoprotein complex, where it's literally the protein incubated with the Fanzor RNA and
Mhm,
like just injected into a cell. Either way, it's very similar to CRISPR in that regard, where you have the CRISPR guide RNA and then you have Cas9 or what have you, and then it's going to go basically search the genome for a region of DNA that's complementary to the guide RNA, in this case
mhm,
the Fanzor RNA, and then it's going to make a cut. Now, unlike Cas9, which makes a double-stranded cut and it just like sledgehammers the DNA, Fanzors make nicks, and a
okay.
nick is basically a single-strand DNA cut.
Mhm,
Now, when it comes to from a cell biology perspective, nicks are generally healthier for the cell for a variety of
mhm,
reasons. When you make a double-stranded break of DNA and it's like a blunt-ended one, like Cas9, so like there's no like sticky ends for the DNA to ligate back together. There's a lot
mhm,
of potential for the DNA repair pathways of the cell to basically just like insert or delete letters at the cut site and kind of like screw up the entire codon reading frame. But
mhm,
when you have a nick, like a single-stranded DNA cut, you can introduce a new strand of DNA without having to worry too much about like the cell like destroying that cut site by like inserting and deleting letters and putting the whole thing out of frame. Uh,
mhm,
and so that's a big one. Like, the being able to make that it's a big feature of Fanzors that you can make these like
mhm,
single-stranded DNA cuts. And that can be an advantage over like traditional Cas9 if we're talking about like
mhm,
a practical comparison. Especially when things get more efficient, that can definitely be more like of an asset for Fanzors.
mhm,
But yeah, so you make the single-stranded DNA cut, and then assuming if you want to like introduce like a new DNA edit, you can basically have a DNA like template that is
mhm,
complementary to the site, the complementary to the region beside the cut site. It'll kind of
mhm,
stick on, and then you can start like you can basically incorporate the edit there. Or if you just want to knock out like a gene, you can just like have multiple cut sites at a, or even like one cut site at a genome, uh,
mhm,
and if you don't provide a new DNA template, the cell will try to repair that DNA, and it can oftentimes make mistakes that'll end up screwing up that region and
mhm,
do what we call a knockout. So, from
mhm,
a genetic engineering perspective, that's kind of how you would use Fanzors, especially as the technology gets more mature.
mhm. My final technical question about Fanzors is that from reading your paper, I know that Fanzors are encoded in transposable elements.
Yep.
So all that means is that there are genes that can jump or move from gene to gene. So how does that change this gene editing process? Does that have an effect on anything?
So, us stressing the transposable elements in this paper is more so to tell like an evolutionary story, because
Oh, okay.
TNPB, like the homologs, are transposon associated genes. They help the transposons, basically integrate into a new site, and so
Mhm.
that's basically us showing that Fanzors are very much like the eukaryotic homolog, like the eukaryotic
Oh,
cousin of TNPB. The founder transposons, and they very likely do, they seem to do very similar things. um and and then our practical demonstration was that you can co-opt the system synthetically, regardless of the transposon features. You can co-opt the system genetically, to genetic engineer new cells, or like genetic engineer human cells.
mhm. So since we're running out of time, hopefully we can do a part two later on. But for today, thank you so much for giving some of your insight on this topic of Fanzors. I learned a lot, and I hope my listeners did too from my high school and other high schools in my city. We learned a lot about Fanzors, how they work, its implications on our lives, and more. A lot of the listeners on this podcast are students from my high school, like I said. So before we end, could you quickly, maybe a couple sentences on advice to high schoolers who are passionate about bioengineering, science, and things like that?
Yeah, for sure. I think it's always really helpful to, do your exploration and learn through side projects. And I think research is a very involved process, and it has, a variety of ups and downs. It can be very stressful at times. And so I think the best litmus test to know if this is like you want, like how you want to approach biology, whether it's through supporting startups in the future, like through the business side, or funding side, or if it's like through a like traditional like scientist path. I feel like the only way to really know is to really get yourself out there and get involved with different experiences both in and outside of the lab. Get yourself into a lab and see if research is your cup of tea. Try following different like following different biotech companies, and if seeing their company developments get you excited, hearing their latest reports gets you excited, or hearing about the latest patents gets you excited, and then also explore that side, and just get a wide breadth of experiences overall when it comes to whatever your passion is, let alone biotech or the life sciences. Try out different things, especially while you're young in high school or even early undergrad. Unapologetically explore, and then start like kind of weeding out the things that you aren't so interested in and doubling down on things you are, and that'll pay off in the long run, because then you'll be able to kind of start early. Assuming you find what you want to do, you can kind of start early and really double down in what you're passionate about, and it'll definitely pay off.
Mm-hmm. Thank you so much for your time and your advice.
Thank you.
Thank you so much for being here, Michael.
No, thank you so much. I really appreciate both the kind words and also the time to read this paper and interview me over it. I'm extremely honored. And once again, thank you for that very very generous introduction. I am, I guess for the audience, I am a normal student like everyone else, and I this kind of research is very exciting, and I think there's so many amazing things to be discovered out there, and so many incredible like research experiences that people have had. And so I highly encourage people to try making your own research experience and crafting your own research story throughout your career, and seeing if it's your thing. If it is, amazing, and if it's not, exploring your interest and finding other ways that you're interested in contributing to the field.
This podcast was surreal for me. Although we didn't have enough time to talk about everything I wanted to ask, I learned a lot from Michael, and I hope you did too. We are going to have a part 2 release in a couple of weeks, so stay tuned. Remember to drop your questions in the comments, and thanks for listening.